Welcome to the Plant Breeding and Genomics Community of Practice webinar talking about mapping thousands of SNPs. My name is Heather Merck and I'm the Content Coordinator for PBG and your host today. For those of you unfamiliar with the Plant Breeding and Genomics Community of Practice, aka PBG, I invite you to explore our training resources at www.extension.org/plant_breeding_genomics. I also invite you to subscribe to our newsletter, PBG News, to stay up to date with webinars and other announcements. During today's webinar, Dr. Alan Van Deynze of UC Davis will talk to us about an analysis pipeline to group, order, and map SNPs, as well as visualize heat maps. Alan is a professional researcher at the Seed Biotechnology Center at UC Davis, and he received a bachelor's and master's degree in plant science from the University of Manitoba in Canada, and his PhD in plant breeding from the University of Guelph, also in Canada. He did a postdoctorate in molecular genetics at Cornell in the Department of Plant Breeding and Biometry. Alan previously worked as a plant breeder for Calgene's Monsanto and a senior scientist for Solera AgGen, where he developed and implemented strategies to incorporate biotechnology into breeding programs. Alan also serves as a liaison between public institutions and the seed industry. And at the Seed Biotechnology Center, Alan's research focuses on developing and integrating genomics into plant breeding for California crops. With Kent Bradford, he co-developed and is an organizer for the Plant Breeding Academy and vice chair of the U.S. Plant Breeding Coordinating Committee. I'll now turn things over to you, Alan.
So my talk today is going to be about mapping a lot of markers, basically, which has been a large challenge, at least in our lab, when we're designing high-density genetic maps. And we've come up with we've adopted basically a strategy that's been developed in the Mitchell-Moore lab here at UC Davis. So Dr. Richard Mitchell-Moore works on lettuce and genomics. Really, Alex Kozik has developed a number of scripts which we're going to, in the programs, along with the advice with the other people acknowledged here, Maria Trucco, Sebastian, Teresa Hill, and Kevin Snowfall, and myself. And we've implemented these in both lettuce and pepper to develop high-density genetic maps. So we're going to go through 3 programs today to really combine the best of these programs. There's the first thing we do, as you know, is we need to group our markers into linkage groups. And Alex Kozik has developed a system of doing this, of Python scripts, and you can see where you can get a hold of that. These are on Unix machines, so the first thing you need to do is really get on a Unix-type server or system to be able to use these programs. And so we group and we do quality control with MadMapper, and then we really align the markers with record. And this is where record the program we really like is record, because it really shines and is very, very quick. And it seems to be quite accurate and compare quite well with other programs like the ones we've listed in our poll. Check Matrix is a nice way of visualizing high-density genetic maps and really looking at ways to troubleshoot and highlight maybe some things that may be out of order or maybe refine maps. So we'll go through all 3 today. The first step is grouping and quality control. The one thing we're not addressing today is really the initial step in data analyses, of course, is quality control in the context of, do you have good scores? For example, we have a seminar or webinar already with Plant Breeding and Genomics on that whole topic for Illumina data. For example, missing, are you going to accept markers that have a lot of missing data, samples that have a lot of missing data, things like that. So we're not going to look at that directly today. So these are things that you need to do prior to this. So MadMapper can group markers. Once you group the markers, you need to basically separate the groups and select the markers within a group. And we'll show an example with, in this case, linkage group 8 that we've done in Pepper. And then we prepare the data and the right files to put those into record. So the input file for MadMapper looks a lot like basically a loc file for most other programs. where you have the lines basically across the top in the columns. You've got the markers in the rows, and you have a header basically telling the program what the data is all about. So you can name the file, the type of population, the number of individuals, and the number of markers in this case. So these files are quite common for most mapping programs. And then the genetic data goes in as A's and B's, and the dashes are missing data points. So for people that are just used to Windows, this looks like a foreign language. But the truth is, if you're going to do high genomics and high density mappings and things like that, you're going to have to address it outside of the shell for most part. And somebody's going to have to type online commanding in a Unix environment for most part. So the program can be called up at the website that I had in the previous slide, and at the prompt you just basically type in these parameters, and I've highlighted in color what each of the options do, and so on. So basically you call the program up, Python MadMapper. You tell it what's your input file, so your loc file. In this case, we've called it test data. You name the file that you want your output to be put in. So in this case, test data out. And then you have a series of options which you need to put in. So in this case, the first one, 0.2, is the recombination cutoff. So the recombination frequency, maximum recombination frequency that you will allow a marker to be included if it's in part of that group. So a marker has to be within 20 recombination units, or 0.2 recombination, for it to be included in a group. MadMapper actually implements another way of looking at recombination through a score that's called bit score. And if you go into the README file and explain how that works, basically the higher the bit score, the higher the recombination, or the lower the recombination, the tighter the markers are linked to. So we recommend a score here of 100, but I don't want to get into what a bit score is. 25 in this case. So this is what's called a data cut. So this is the minimum number of observations that the program will use between two markers for them to be included in the group. Okay, so you have to have 25 markers that you have data on for that marker to be included in the group, basically. If you have already a map that you've been using and you have a framework map, you can list which markers in your data set are considered framework markers. So these are usually high-value markers, or markers that map with good confidence and so on. The allele, what we call the allele distortion, 0.33 in this case. So this is the maximum allele frequency minor allele frequency that you will allow for a marker to be included. In this case, we're doing recombinant inbred lines. You want 50-50 would be the optimum, but we're allowing some leeway up to 0.33 in this case. Missing data. In this case, so the number 50 here for missing data actually is the number of missing data points as opposed to the percentage of missing data points. So this number, of course, will change depending on how large your population is. So this is the minimum number, or the maximum number of missing data points you're going to allow for a marker to be included. The trio analysis is basically what's called triplet analysis. So you can do pairwise analyses for grouping, or you can do triplet analysis for grouping. For first pass, we really want to just do a pairwise analysis. So we just put in no trio. If you look down here, so your two options are trio or no trio. You want to put no trio also because it's much, much quicker, because then you're just looking at pairwise comparisons as opposed to all possible triplet comparisons. And you can put in a number of double crossovers, a maximum number of double crossovers that you're going to allow in the mapping or the grouping process. And we recommend 3 in this case. So what I do want to say is all this is in the README file under the MadMapper when you click on the link, you can download the README file, and it explains all these actually in actually more detail than I have even today. Oops, I went too bad. So you run MadMapper. You basically run this script. You hit enter, and if everything works out and your files are all correct, MadMapper quickly generates 76 files for you. And we're not going to go over 76 files. The main one you want to look at this time for grouping is called the tree cluster file. So it's going to give automatically puts out the output file name that you defined in your script. So test data out, and then it adds this xtree cluster file. And you can grab that file and you open it, and you can open it in Excel, and it basically is an Excel file in Excel format. And this is what you see. You see an Excel file with a number of headings and a bunch of information. So at the top you see 0, and up to this case the 0.2. So this stops at 0.2 in this case because we've said we're not allowing anything beyond 0.2 if you remember back in our options. So 0.2 recombination. And what you see underneath here are basically the number of groups for the markers, which are down here in column C, that you have if you have a 0% recombination and so on. So what you really want to see is that you get to 0.2, you want all the markers. So if you read this correctly, all the markers from, say, line 26 to line 32 are in group 1, actually down to a 0.18 recombination frequency from that. So as you get tighter, you get less of these markers, and these are basically group numbers. So if you look at group 2, you see the same thing. Group 2, these markers are really very tightly linked. These are actually all in the same genetic bin if you want to look at it, because there's 0 recombination up to, you can slide over and you can decide, so at 10% we have, these markers are all together. At 20%, basically all the markers are together. The one thing I want to put out, we don't usually keep, say, this bottom marker here, because it's kind of on the edge. It's a point, this marker is part of group 2 at 20%. And sometimes you'll see a number of markers down there, and those, you might question further depending on the genetic distance when you look at things later. So what you need to do is basically, so now you've defined groups. You have group 1, group 2, and group 3, and so on at the 20%. But if you don't like 20%, you can make that cutoff anywhere you like. If you say, I'm really interested, I want to be more stringent this time, I'm going to go at 10%. But so you have all that information in front of you, and you can decide how you want to make your groups and how you want to break those groups apart. So you see a couple more columns. I'm not going to get into a graph class of node type R. These are really just parameters that define how well things are grouped together. The stars are just really breaks for the output. In this case, we have framework markers for group 1 in this example. So this marker over here, number 246 and 126, we've seen before, and as part of our framework, and they were in our framework map at position at 12 centimorgans and at 20 centimorgans. So these are basically anchors and references that you can put your new markers towards if you have an anchor map already and that you want to use. And you have your markers, you have a count of the number of A's, the number of B's, and the total number of individuals in your population. So this is part of the Excel file that you see that's put out in this cluster file. So the next step that the next thing we want to do is really now add the data and make a subset or a loc file, basically an input file for record with the actual genetic data, with the A's and B's associated with each of the markers in the groups. All I've done is I've slid the file over. I've just scanned the file over, the cluster file over, and now I see this part. for it. So these are the markers. These are all in linkage group 1. So we've added linkage group 1. We've just added this column here in Excel, just by putting LG1 and putting it down to where the breakpoint was for linkage group 1. And here are the markers. So now we need to add the data. And so what we do is we query basically the loc file, our original input file that went into the test data file, that went into MadMapper initially. So it has all the genetic data, of course, the A's and B's. And you can use a function called VLOOKUP. What VLOOKUP does is, and this is a standard function in Excel. So we put this function, we type this function right here, and you just pull the function down, and you have a number of arguments. So he says you look up at the value. In this case, so the value is the marker. So Z2. No, you can't see it depending how big your screen is. So we point to where the marker is, and then we point to basically the Excel file that has all the data. Okay, so in this case I have it in sheet 1, which looks like here, and I've highlighted the whole range. So I highlight the whole sheet where the data is. So A2, in this case the D2, there's 6,000 markers in this data file, 6,000 lines, 599, and so on. And there's an index column which basically tells VLOOKUP program start over here, and you just start at the top, A1. The false in VLOOKUP means I want a perfect match. I only want data exported if I only want the data exported in this file if I have an exact match between this marker name here and the exact marker name in that file. So you hit go, and what that does is really populate all the genetic data to the right. So it takes this as a reference, grabs the marker name, and basically copies everything for this marker that is in the loc file. So what you've done now is you have a file that looks like this, which is basically looks like a loc file, but you have now all the markers that are in linkage group 1 in this case, and its genetic data. So you have a new marker, a new file for each linkage group that you have. So basically what we're doing is we use the group data, which transferred the genetic data, and we're going to save each group, the genetic data for each group, into its own file and make a loc file, basically an input file for a record by group. The next thing you can do is, if you want to do a trio analysis, you basically use the same MadMapper script as we had in the past, except you change the word trio, and you do it, your input file is by linkage group, in this case linkage group 8. What the trio file does is give you a little tighter grouping, and it gives you a few other statistics like allele distortion. It basically puts out a few extra statistics on the markers, and it can give out, then you can really pull out a subset of the markers within that group that may have a higher quality than normal. We actually don't use this trio analyses in our analyses. The second thing we need to do within MadMapper is really export the pairwise matrices, which we'll use in further analyses, such as check matrix. So again, we use the same script for MadMapper as we did initially for all the markers, but our input file is now our linkage groups, in this case linkage group 8. So it's exactly the same as before, but now we have, we did this in Pepper, for example, we have 12 linkage groups. We're going to do this 12 times, once for each linkage group, or if we have more than 12 linkage groups, we do once for every linkage group. And then all you do, put in the exact same parameters and options, but you change. Of course, your input file is different, and your output file is different. And that's all you do. So it automatically will generate, when you hit go, the pairwise matrices for each linkage group. And so again, so each linkage group input file looks like this when you're done. So we've grabbed that file, that plus file, for each linkage group, and all we've done is added the header. the header. So linkage group 8 in this case has 150 markers instead of the 6,000 that we had initially to do that. Same number of individuals, of course. So the one thing that we haven't figured out to do, and if this gets you back, is to rename files into the log file. So to rename your in Excel you would have saved your file as a text file, but you have to change the ID to .loc. And you can do that easily. Just go back to your DOS command line in a PC, and basically go to your directory and just use the word rename, the command rename, REN, and then the file lg8.txt, which is the name you've given it when you saved it from Excel, to lg8log. So basically this is how you've renamed your file and is your input file for the MadMapper, and also for record. So record is a program that's basically come out using from this website from Wageningen. And if you click on here, basically it tells you a little bit. So you go to this website, this is what you see in the page, tells you a little bit about the program. And you can download two different programs. There's Record, this is Record for DOS, and this is Record for Windows. They do the exact same thing, except Windows, of course, has a shell type program. Today I'm going to talk about the Record for Windows. And there's a couple differences between the two programs, which I'll highlight. So if you download the program for Windows, and that really takes about 2 minutes, it's very, or not even, it's very, very quick. It's a simple program. You get an opening screen, and it tells you a bit about the program, the types of populations it can. So what does it do? It maps markers for a single linkage group. And these are the type of populations it can use: backcross, F2s, recombinant inbred lines. You can, of course, use double haploids under the backcross command, but your distances are overestimated from the backcross. And basically, the way it works, like most mapping programs, it tries to minimize the total number of recombinations. Record has basically the NST map program uses Record, the same exact algorithm as Record, just to give you an idea. And you can even export Record results into JoinMap and other programs if you like. Okay. So you open the program, this is what it gets. It really only does 2 or 3 things. You have to start a new project. You load the data, and these are, you load the data, so you can actually capture load from file, or just hit this button over here, load data. And this is the log file. This is the log file for Group 8 that we created. Here are your markers. Here's the genetic data, the header. So nothing new, the exact same file that goes into MadMapper and JoinMap and other type files. So the first thing, so you go to your ops. So the next thing is options. So really, what's really nice about this program, the way to use it is you just go through the little icons over here in that order, and that's what you do. And so you hit the options command, and this is what you're going to see. There's very few options. The first one is what's called the maximum additional recombinations when seeking plausible alternatives. This is not the minimum distance to include a marker in your genetic map. This is the minimum genetic distance or recombination between alternative genetic maps. So in this case, so what it does, it goes through a number of iterations of the map, in this case 5, and these 5 maps cannot be more than 10 centimorgans. The total distance cannot be 10 centimorgans apart. Okay, so it's looking for a resolution of multiple maps and give you the best map. The gap size, so this is maximum recombination allowed between 2 markers for them to be included, is 25 centimorgans, and you can choose Haldane or Kasambi. Go back to your menu, and then you can hit calculate over here or in the calculator, and then you get a whole bunch of output. And I'm going to go through quickly what you see from the output. And you can see the output either in the log file or this file is automatically saved to the directory that you set up your project in as well. And it's called JM order. I think it actually means join map order, but because it's the same developers. But you'll find a file called JM ord, and it's basically a text file and has the exact same thing as your log file. So at the top of the file, it'll show you kind of the options. So this is what you've seen in the options. You had a maximum alternative recombination 0.1, 5 steps, Kasambi 25 centimorgans maximum. And basically you see a list of the markers that have, what's nice, it says it lists a set of markers that have the exact same basically haplotypes, or basically genetic segregation. All right, so it's not going to try to, and what it does, it just chooses one of those. So it doesn't map all your markers at once in initial steps, and so on, just to save time. And this is really important once you get to really high density maps. I'll give you an example. In Pepper we map 16,000 unigenes, but we only have 2,800 unique haplotypes, or 2,800 genetic bins in that map. So it's a much easier task to map 2,800 than it is 16,000, of course. And then you reassociate after the fact the haplotypes that are the same. It goes through 8 different replicates, and the last replicate is the best one, and it gives you the total genetic distance. These should be within 0.1 centimorgans, basically, which is your value. In the notepad, and this is truncated, it gives you the best estimate of the order. And here's your order from 0, in this case, to 149. We had 150 markers. The total genetic distance, in this case, 160 centimorgans. And it gives you the genetic distance below this. Okay, so here's the exact same order of markers, and here's their genetic distance. So these markers are in the same genetic bin. They're all at the 0.417 distance in the map. So the DOS version basically has the exact same commands, but it's much, much quicker. What can take maybe 100, this subset of data set might take maybe 10 minutes in WinRecord, or sometimes an hour with a large data set. It takes literally minutes in DOS. With really, really large data sets, basically DOS is minutes. You can go in hours with WinRecord. But it's not days like some of the other programs like JoinMap, for example, in the same data sets. Really have taken us overnight or days to do the same linkage groups that we've done in record in less than an hour. And so this, and especially with the DOS, it's really minutes. The thing though the DOS version does not do, it does not export the distances. Okay, so just export the order part of the file that I just showed you in the previous slide. And so with that order, you can get the genetic distances by importing that order into JoinMap, and then JoinMap is actually quite efficient once you give it an order, because then you're just asking it to calculate the genetic distances based on the order you already have. So that's something we've actually tested between the two programs. In the genetic order, the order is pretty much the same between the two programs, except for very, very close markers, less than half a centimorgans, where honestly you don't really have the genetic resolution most of the time to make those calls in the first place. So the next step is let's look at the genetic map and qualify it. And Alex Kozik has come up with a nice program called Check Matrix, and it's available at the MadMapper site, the same site that we started with. And it needs 3 sets of files. The pairwise distance file, which we generated in MadMapper when we were doing the groups. It needs basically the order of the markers from, and not even the distances, just the order, in this case from record. And I say reformatted. Basically all we've done to reformat it is we've grabbed the marker names, the orders, and we've added a column with the linkage group number here on the side. Right? And so we've added one more column with the linkage group number. So you can collate all your linkage groups to run through Check Matrix. And then the log file, the same one that you put in the input record, to start with for your linkage groups. Then you run check matrix. So check matrix, again, you have a Python script. So you call the program up using this this script here, basically Pi matrix, and you call up the pairs file from MadMapper. You call out the order file, which we just exported from record, and you tell it the name of your output file. The X and the Y, in this case, are just basically dummy variables where if you do have a framework marker list, you would put the name of that file with the framework marker list. If you actually have a list of markers that you want to highlight in red, or basically highlight, in this case it's going to come out in red, for any reason. They could be key anchor markers, they could be markers for traits that you're interested in. It really doesn't matter. Any markers that you want to list in red. Your input file, basically your for your linkage group, and then basically, do you want the graph to be displayed based on recombination, bit scores, or LOD scores? If you go down here, those are the different options. So you could put rec, bit, or LOD here. Check matrix can put out a type of graph. It's really a circular graph that it's like a network graph between all markers, which we don't use generally, so we just put in the option no graph in this case, but you can put graph or no graph. This is the maximum recombination or the linkage cutoff. We just put this at a maximum of 0.9 because we already have done this in our grouping in our record analyses. Okay, so we just say include all the markers. We don't really want it to be filtering our markers at this point because check matrix is really a visualization tool as opposed to a mapping tool per se. Small, you can put small or large. This is the size of pixels, and you'll see what I mean when I show what the check matrix displays look like. It's really the size of pixels you want to see in your display. We use small. If you have a lot of markers, you would probably want it small as well. And again, the population type. And again, all this is explained in the README file for check matrix once you click onto the line, online to it. The next thing you want to do is to look at your file on either your PC or your Mac to display it, and you have to be able to grab that file from your new Unix system, and bring it to your computer, bring it either to your PC or to your Mac. And there's different programs that you can use. One is WinSCP. Here's the download for it. And what it does, it basically opens a window and basically allows you to see the directories and the files you have on your Unix system here, in this case on the right, and the files on your computer on the right. And you can basically drag and drop the files you want. The file you want, so you find your check matrix folder, are the PNG files. And those are basically the files or the diagrams that check matrix puts out to let you look at your maps. And this is exactly what you see for check matrix for a single group. And there's a lot of information here which allows you to see thousands of markers in one look. So what you want to, basically what check matrix is doing is aligning all markers by all markers. And it's asking the question, are they genetically linked? And basically what you want to see is a nice red line down the middle over here. So basically red means high linkage, strong linkage, and blue means negative linkage. You really don't want to see blue within a linkage group because it means there's some markers that really are in repulsion to other markers within that linkage group. And the yellow, and basically, it's grades of color from basically red to blue. So there's a number of things you see here, although it can be fuzzy. So the areas that have large spots, I mean there's a lot of markers in this area that are tightly linked. So areas really of low recombination, and where basically you see, sometimes you'll see a very constriction of the graph here. You see, this is not a very, you'll see finer constrictions in further graphs that I'm going to show. These are areas of high recombination. essentially. And you can actually see each of these pixels lines up directly to the marker over here on the right. And you actually have your genetic map over here on the right as well. So the area of high recombination, if I slide over to the right, and you can zoom into these, onto your screen without losing too much resolution, which is nice. So you can zoom into this area, you go across, and you see there's more recombination in this area than there is in the area over here with low recombination when I slide over to the markers over here. Okay? So again, this is a nice, if you want to call it 10,000-foot view of your genetic map. The other thing that's nice on the bottom, it shows proportion of A and B alleles. Basically, you're looking at distortion over here or no distortion. So what you want to see is it's an even, you want to see nice even red and blue contributions in the recombinant inbred line population. So you don't have, so it's your allelic proportion or you can call it distortion. And so the yellow here is showing there's a little, for all these markers, there's a higher frequency of the A allele, a slightly higher frequency of the A allele than there is the B allele down here, and so on. And in this case, there's a little slightly higher frequency of the B allele than the A allele. So that's what the kind of the lighter blue and the yellows are showing over here. And so this is one part of the graph. If you actually slide to the right, you actually see this next part of it, and you see the actual graphical genotypes. And you can pull these as single images or as images with the previous image. So here's your graphical genotype, which also shows, again, your markers, their order, the genetic distance, and also the allele frequency, just like before, in just like a different graphic. The other thing that you see here, it's not that easy to see in this case. You see gray and white, white little pixels, essentially. The gray pixels mean there's missing data. And what you're seeing over here, there's 4 lines that we chose to leave out of our analyses. So we put them all as missing data. Because there's a number of reasons to do that. Maybe it's just because they have too much missing data, or basically maybe they have too many heterozygotes in this recombinant inbred line population. But we're not comfortable in keeping these lines, 4 lines in our data set, because it's kind of messing up our analyses. And so you can just cancel these guys out. But of course, there's individual missing data points for different markers for different lines. What you're seeing in the blue and the red here are the double crossovers in there. So you want to minimize double crossovers, of course. And here it counts the double crossovers at the bottom. You have 2 rows, but that's just really for display. So this is really line 1, line 2, line 3, and so on. So in this case, line 3 has 4 double crossovers across this linkage group and so on. The X's in this case show that, this guy has a lot of double crossovers. Maybe there's some problems in the markers order in this area. If you go across, these markers actually represent areas of low linkage, and sometimes you basically don't have you may have double crossovers in the order here, but you may not also have the resolution to make that call in the first place. And so you have to balance that, as a scientist to say, are these really a problem, or are there areas that the markers are so tightly linked that we really can't make much, really not sure of what that order is. So I'll give you an example of how we've used that tool in Pepper. So we used our Pepper Affymetrix chip to, again, we mapped about 16,000 different unit genes on this. And this is a genetic map. So Pepper, we mapped them on a interspecific cross where we knew that there's a translocation between chromosome called P1 and chromosome H between the wild type and the cultivated type. So what translocation does, it basically gives you false or pseudo linkage. And this was elegantly actually shown in a Pepper map actually created by Wu et al. Out of the Cornell group a few years ago, 2009. And so they identified this, I'll say, with a relatively, what's the word? This is not an ultra-dense genetic map, but it really captured the translocation nicely. So what you see is a reciprocal translocation where P8 while, the section over P8 while over here is actually translocated to the bottom of chromosome 1 in the cultivated peppers. And at the same time, there's a section of the cultivated P8 which is found in chromosome 1 in the wild type. So basically the P8 basically is translocated down here to the wild type. So what that does is give you pseudo-linkage between chromosome 8 and chromosome 1. And we saw this really nicely in check matrix. So basically what check matrix is saying is the markers in this area have pseudo-linkage with the markers in this area. So that's why you see these dark red dashes over here. So this is not what we want to see in our genetic maps. And there's actually a very nice breakpoint here which basically says, you know, this is a big gap. There's a lot of recombination. Here's a probable area where this linkage or the pseudo-linkage exists. So this is basically a mistake. We've put together P8 and P1 together in our genetic map. But we saw this right away. It just glared out to us and said, okay, we need to break these linkage groups. We did checkmate our first round of mapping with check matrix. This is what we found. We compared all markers. Here's the linkage group I just showed you before. So these, but we got 9 linkage groups. We did this initially with lettuce, and with 9 linkage groups in lettuce, we were ecstatic because that's how many chromosomes are in lettuce. But we have 12 in pepper, so that's a problem. And we found basically 3 areas where there were some logical breakpoints where we have these really fine areas of basically repulsion. If you want to see, you see these blues all over the place. So this was first the P1, P8 translocation. And there was 2 areas right here also in linkage group 6 and linkage group 7. And you see this repulsion saying, you know what, the markers over here are really not happy with the guys down here. And so we broke those linkage groups, we refined our map, and this is what we get. We got a really nice map, genetic map. Got our 12 linkage groups, about 2,900 genetic bins, and actually we're writing up the publication for that right now. But basically what I wanted to show with check matrix is that it's a really nice way to find, you know, to look at your linkage groups, your genetic maps, and really refine them and pinpoint maybe some problems that you have. The other thing that's really nice is that each pixel is connected to a marker. You zoom in and you say, ah, this is a marker that has a problem. I want to go back to here. There's a couple other highlights. You see over here there's this yellow versus everything around here is red except there's a marker here that is yellow. So it kind of belongs here, but for some reason it's not as tightly linked as the other markers. So we can go to this marker and ask the question and look at the data again and say, you know, is this data good? Where does this marker really fit in here? So it's not just the, you know, the glaring breakpoints that it shows. Everywhere there's basically a break in the color, so to speak. You ask the question and go back to the marker order and see if you can refine it. And so we did that, and again, so we've minimized. We don't really have any of these glaring breakpoints in this genetic map, and we're really quite pleased with this genetic order. And we verified this genetic order in join map, and we got basically the same results. So what did we do today? I know I'm speaking fairly fast. You know, we used MadMapper to group markers. We basically massaged the data and got it. We broke it up by linkage group using the VLOOKUP function in Excel. We created a paired matrix file for each of those linkage groups, and also a input file for a record by linkage group based on the groups. And we've ordered the markers in record, and you can do that again in DOS or in Windows. Windows will give you the order and the distance. And again, the genetic distance between join map and record in the Windows version is really quite close. The DOS version, you have to get the genetic distance. You can do that by importing the join map. And then we've shown you how you can really look at your genetic maps, ultra-dense genetic maps, and really troubleshoot them and really refine those. And with that, I'm happy to take questions.
Well, thank you very much, Alan. I think you gave us a really nice demonstration of sort of incorporating some different programs to really deal with large data sets. And we have now about 15 minutes to answer questions. And I would just like to remind you that we cannot take verbal questions. So you'll need to type your questions into the question box. And to do that, you just type them into the question box. If it's not visible, you can press the little plus sign beside the word question, and it'll open up your question box for you, and I'll read out the questions, and Alan will answer as many as we have time for today. And to start off, we've had a couple of questions, Alan. I wonder if you can just comment if these software programs are publicly available or freely available.
Yes, all three software programs are freely available. Just click on the links in the first couple slides, and then you should be ready to go. The catch is, though, you do need to be working on a Unix system for the MadMapper and the check matrix software.
Great. And the next question here is asking about what types of markers this is suitable for. So can you use something like microsatellites?
These markers are really good for, these programs are good for any type of markers. Basically, if you put them in A's and B's, relative to, the parent genotypes, that's all it requires. So whether that's a microsatellite, a SNP, a HRF-LP, anything you like, as long as it looks like the input file, you're ready to go.
Great. And I don't know if we can go back through here. Sorry, seeing a lot of questions pop up at once. One question is looking at. There's several questions looking at populations that can be used. So a specific question here is, can you use backcross 2 or more advanced backcross populations?
Well, basically, the MadMapper addresses the same populations really as record, backcross, recombinant inbred, and what am I missing? F2s. And so whether that's a backcross, 2F2 or backcross 2, the only difference between, if you don't have populations that are defined exactly like that, all you need, the difference is really your order is not going to be your order will be the same, but the genetic distance will not be accurate. So depending on the population, your genetic distance might be over or underestimated, depending how you define it. Which is typical, and this is not specific to, to record or any other program. To these programs, that's the case with all mapping software.
Right. Yes. And a related question here, and a comment. First of all, thanks for a very nice webinar. And is record effective with cross-pollinated population structures, like the CP and join map? I'm sorry.
Is it effective in cross-pollinated? Well, again, it goes back to population type. It can handle. It's basically diploid genetics. It's based on diploid genetics. F2 backcross and recombinant inbred line populations. So it's limited to that. It really doesn't try to go beyond that as far as populations that may have not those typical segregation expectations. Mm-hmm. So it certainly doesn't do tetraploid analyses like our last webinar, basically last week on potato.
Mm-hmm. Great. Yeah, I think that's what this person may have been getting at here.
No, it doesn't address that.
Great. There's a couple questions here about the input file format for MadMapper, and I can't remember if you showed the input file for that or not.
No, I did. A couple times. The input file for MadMapper is the same as record, and basically the same as join-map. I showed 2 slides where I had basically the genetic data with the lines across the top of the markers on the markers as rows, and where you're just defining a file, basically the file name, the type of population, number of individuals, number of markers, with a header at the top.
Sure. So it sounds as though it's a pretty standard
Exactly.
file format.
Then. Yeah, you can basically use the same file for join map.
Perfect.
With the exact same file.
All right. And there's another question here. If we can create integrated maps at all, or if this pipeline could be adjusted to address some integrated mapping.
It's not something we've done. I mean, it wasn't set up for that at this point, no.
And is there a cutoff for the amount of missing data that's acceptable by the software?
I don't think there's a cutoff, but there's probably a functional cutoff where, you know, if you have 70% missing data, of course you're not going to use it. But there's a point where all your linkage groups aren't going to make any sense anymore. So the results are going to basically tell you that you've got a problem.
Mm-hmm.
So I think the program will function, but your results will be desirable.
Right, and you're going to need to use your judgment and your insight to be able to
exactly.
And you know, a little bit more of, I think, sort of just a thought-provoking question, or a little bit different track. One person's asking, why do you want to map 10,000 plus markers?
So I don't know if well, this is but right. This guy's a plant breeder, I know it. Well, the reason we're doing it is really we're looking at trying to use these for QTL analysis and really to look down at candidate genes, for example. There's a number of other reasons on a genomics level. For example, you could use these type of maps for anchoring genomes, and where you're really starting to look at, again, starting to work with the genes as opposed to just the markers. You know, and the idea is to get markers that are really the genes that you're looking for, and start making answer biological questions, really, as opposed to in breeding, where you just, in a sense, want the marker that's linked to your trait and kind of use it routinely. So that's our reason, anyway. We're looking for candidate genes and really looking for genes controlling traits and try to get a better handle. And what we've found, and there's some nice examples of programs also, when you start working at the genomic level, even in breeding, you can start and understand really what genes and what traits, how traits are really being developed in your breeding program. Or you can start designing new things and really start breeding in a different way.
So we have a comment here that I believe may be from someone who's worked in the development of this software, that says, record can do grouping as well. It needs some tweaking by allowing a gap of 50% recombination. From the output, you can deduce the boundaries between linkage groups. You can also load the marker order in Excel and create graphical genotypes and visually decide the boundaries of linkage groups. And so this could, if you wanted to, help you avoid using Python scripts.
Okay, that's a nice comment. Thanks.
Yeah, so thank you very much. And as a follow-up to the question about mapping, say, more than 10,000 markers, the question or comment here is, if you want to separate 10,000 plus markers, let's say for each candidate gene, wouldn't you need a very large population to see any recombination between those tightly linked markers?
The answer is yes. So we never, I think, what I alluded to, we never really used record to map, say, 10,000 or 16,000 markers. We actually, before we added our markers, we looked at the common haplotypes. So basically, we only chose one marker out of a group of markers that had the exact same segregation, and then we input those, and then you just reassociate the markers into that genetic bin. So basically you're mapping genetic bins. What's mapping all these additional genes that that are not recombined, for example, within a genetic bin, these just become candidate genes, which then you need to follow up with further populations where you want to zero in. And you know, you can follow up with further populations, you can do transformation, you can do all sorts of ways to try to validate those candidate genes. So basically it falls into a program. It's not the only tool that we use.
Mm-hmm. And a question here. I know you looked at the Kosambi mapping function. Is there another function used to estimate recombination frequencies?
Yeah, Haldane, which is an option, of course. And the difference between Haldane and Kosambi is Kosambi function basically has a factor in it to assume that there is interference between closely linked markers, whereas Haldane does not.
And we have another question and comment here. This person says, I think the question about outcrossers refers to self-incompatible species in which inbreds and classical F1 and backcross 1 populations are not possible to produce. Crosses are usually between parents with a high degree of heterozygosity. It does not appear that the program would handle these populations, sometimes referred to as a pseudo-double test, pseudo-double test crosses. Is
Right.
this correct?
I believe so. I think that's correct. This program doesn't address it. It's basically, this program is looking for cases where you only have two alleles, you know, one from each parent.
Mm-hmm. And another question here, wondering if you've tried to integrate linkage maps that you've created in JoinMap into your pipeline, or if you thought this might be an easy thing to do.
It's not easy. But, you know, JoinMap will do that, of course. So you can, I have not done it, but of course I think JoinMap would be an appropriate program to try to do that.
Great, and it looks like we just have maybe 1 or 2 more minutes if there are any remaining burning questions. So let me see if I can. So it doesn't look like anyone is typing any last-minute questions in here. So with that, I'd just like to. Oh, we've got one question here for you, Alan. Have you tried MST-Map?
I have not. We did. I personally have not. A colleague of ours has, and they have compared MST-Map with record. It's the same algorithm, so you get the same results, or very similar results.
Great. Well, thank you very much, Alan, and thanks to all of you for a nice discussion here at the end and some very positive feedback. We very much appreciate it. And as a reminder, today's presentation is available by following the link provided on the screen. We also encourage you to sign up for PBG News. That's our newsletter so that you can hear about new content and webinars. And we hope that you'll join us again for future webinars. Thank you very much and have a nice day.